In this video, we will cover multiple steps related to visualizing gene expression and identifying spatially variable genes when analyzing Visium spatial transcriptomics data in Chipster. We will talk about visualizing gene expression and identifying spatially variable genes with multiple tools. The first tool computes and visualizes spatially variable genes based on pre-annotated clusters. The second tool identifies and visualizes spatially variable genes without pre-annotation. Finally, we show how to visualize gene expression of selected genes on top of the tissue image. When we are trying to identify spatially variable genes, we are trying to find genes whose expression correlates with a spatial location in the tissue. The first approach to identify spatially variable genes is to use pre-annotated clusters of anatomical regions to compute differentially expressed genes. This is the same approach as in the single-cell RNA sequencing data analysis workflow when we compute differentially expressed genes between clusters. But here, we also visualize the differentially expressed genes on top of the tissue image. This method works well when the clusters, as shown here, show clear spatial restriction. The second approach does not require pre-annotated clusters. We offer two methods for identifying spatially variable genes without pre-annotation. You can either use the marked variogram or the Moran's-I method to identify the spatially variable genes. The Moran's-I method is the default method and it's much faster to run. It measures spatial autocorrelation, so how much do the nearby locations resemble each other in terms of gene expression. The marked variogram method models the spatial transcriptomics data as a mark point process and computes a marked variogram. The variogram is a measure of the dependence between the marks of two spots that are a certain distance apart. As you can see, there can be differences between the identified genes, so you can also try out both methods. Now let's take a look at how to use these tools in Chipster. If we're identifying spatially variable genes based on clusters, we can use this tool and choose the clusters for this step here. We can also limit testing to genes which are expressed in at least this fraction of spots, as well as limit testing to genes which show at least this fold difference. We can also choose the test for testing differentially expressed genes. And we can also choose whether we want to report only positive marker genes. We can also choose the number of spatially variable genes we want to visualize and whether we want to determine the color scale based on all genes. And then we can run the tool. This tool outputs visualizations of the identified spatially variable genes, as well as a table of the differentially expressed genes between the clusters. If we're identifying spatially variable genes without pre-annotation, we can use this tool and choose the method we want to use here. We can also choose to lower the number of variable genes used. We can also choose the number of genes we want to plot and whether we want to determine the color scale based on all genes. By default, the color scale is determined for each gene individually, and this may differ between the genes. We can also choose the same color scale for all of the genes if we're comparing gene expression between genes. We also need to specify if we're performing multiple sample analysis, and in this case, we're not. Now we can run the tool. This tool outputs a list of spatially variable genes, as well as the visualizations of the identified spatially variable genes. As you can see, the color scale is the same for all of the genes, which makes it easier to compare the gene expression between the genes. If we're interested in visualizing gene expression of specific genes, we can use this tool and choose the genes we want to visualize here, as well as select other parameter values for the visualization and run the tool. And here are the results for this step.